All right, how's it going? Back with another talk for you. This is on a new topic. We're still dealing with cells, but now we're going to talk about cellular respiration. Sometimes we'll also call this metabolism. Those terms are often used interchangeably. I'm going to split this into a couple of PowerPoints because we've got quite a lot to cover. So let's get started. Okay. So what's the importance of understanding cellular respiration? Well, without it, nothing exists, right? Life exists because we can capture, some organisms can capture sunlight energy. The rest of us can harvest that energy from those other organisms. And understanding these cellular reactions that are a part of metabolism gives us a more deeper understanding of these organisms. Also, many of you are going to be going to med school or vet school, and so this is where a lot of medicine occurs, is understanding human physiology and the things that go on in the cells. But also, I like to talk about this subject because it introduces, again, a lot of basic concepts that we're going to use throughout the semester. And so some of those basic concepts, the laws of thermodynamics, enzymes, we're going to be talking about enzymes all semester, ATP, and finally cellular respiration, tying it all together. So let's get to work. So a broad definition of energy is, of course, the ability to do work. And then you can talk about energy in different ways, and depends upon what we're talking about. It depends on what class you're taking. Basically, we've mentioned kinetic energy before when we talked about temperature. Kinetic energy is the energy of motion. It's the energy that moving objects have. Potential energy is stored energy. And so one example of this would be a boulder sitting on top of a hill. It's got stored energy and it wants to roll down the hill. But the example that we use a lot in here is chemical energy. We've talked about how when you push those atoms together, it takes energy. When they bind to one another, that energy gets stored in that bound, and so then when you break that bond, it releases that energy. And animals convert this potential energy to kinetic energy. And so we use this chemical energy and convert it to moving energy, whether it's making molecules move around, making molecules change shape, other reactions that make the organism move around, whatever. We'll take some of the potential energy and store it in other molecules, and so we convert it from one form of potential energy to another. But ultimately, you're going to use a lot of that energy and turn it into kinetic energy. Okay, so we've mentioned the laws of thermodynamics before, but they're kind of important, and so we're going to talk about them again. The first law of thermodynamics is that energy is neither created nor destroyed. It just changes forms. And so you never make energy. You never make energy from anything. You just capture energy and turn it into a different form. All right? And that's it. There's only so much energy in the universe. The second law of thermodynamics, we've mentioned this before, when we talked about random molecular motion and things tend to go from a high energy state to a low energy state. Another way to word the second law of thermodynamics is that all of these energy transfers are inefficient. So you can't make new energy, you just change it from one form to another. That's the first law. When you change energy from one form to another, you always lose a little bit to heat. That's the second law. And eventually, all the energy in the universe will all be converted to heat. That's called the thermal death of the universe. Not anything for us to worry about in the immediate future. Another way to mention this is that all energy transfers increase entropy. So entropy is another word for randomness. And this relates back to our earlier definition. So things tend toward disorder. Then things tend to go from an ordered state to a less ordered state. That's a natural law of the universe. So to get things in an ordered state, you have to put energy into them. To get things to a higher energy state, you have to take energy and put it into that organism. And so, you are an ordered state. You are lots of molecules that are very ordered and coordinated. That doesn't happen spontaneously. It only happens because we get a constant influx of energy from the sun. And that's why we can have living things on this planet. The disorder of the solar system, the disorder of the universe gets greater as those reactions take place in the sun. And so on a very local scale, we are getting more order and a higher ordered state, but the expense is the overall universe is becoming more disordered every day. And so again, this figure from your book, just reinforcing what we've said several times, solar energy is captured by the primary producers and converted to chemical energy. And then it gets passed up the trophic chain as the herbivores eat the plants, and then the herbivores get eaten by carnivores, and those carnivores get eaten by other carnivores, and so on. That energy that sunlight energy that was captured and put into those bonds gets transferred and gets changed to other chemical bonds, but each time you lose some as heat. That's the second law of thermodynamics, which means each time you have less and less energy. Each trophic level has less and less energy because the trophic level below it has already used some of that energy and converted it to heat, and heat can't be used for much, and heat cannot be put into a chemical bond. And so that's why we only have 3 or 4 trophic levels. I say that if you're walking across campus in Murray, you're not afraid of anything. You're not afraid of getting eaten because we're at the top of the trophic level. There are no trophic levels higher than us because there's not enough energy in our trophic level to support another organism. Now, in certain areas there is. If you were somewhere where there were grizzly bears or something like that, but the grizzly bears are not eating just humans. The point being is that every trophic level has less energy stored in it than the trophic level below it because each trophic level uses heat as it's converting energy. That's the second law of thermodynamics. Okay. So when we talk about this energy, we're going to talk about the energy that gets converted as we break these bonds, right? We've said you put that energy into a chemical bond, then you break that bond and release that energy, and then you can put that energy somewhere else or do something with that energy. And so this is a diagram we're going to keep looking at time and again. It's figure 4.3 from your book. And on the x-axis, you've got the reaction process, so that's just basically time. And on the y-axis, you've got internal energy, the energy of the molecule that we're working on. And so what this is showing is that your molecule has some energy stored in it. That's why it doesn't start down at the bottom of the x-axis or the y-axis. It starts in the middle of the y-axis because that molecule has energy. Then if we're trying to break apart that molecule to get its energy, we have to add energy, and then the molecule breaks apart and it releases a tremendous amount of energy. So we're going to talk about these steps in more detail. So again, we start with the chemical energy that is stored in the molecule that we're going to break apart. Where did that energy come from? Ultimately, that came from sunlight. This is, a photosynthetic organism captured some sunlight, took that energy and converted it to chemical energy. And then as that plant or whatever got eaten by an herbivore, that energy gets transferred to the herbivore, and then that gets eaten by a carnivore. Each time molecules are being broken apart, the energy is being transferred to other molecules. And so ultimately, this molecule has energy in it because originally that energy was captured by a primary producer. Now we want to break apart this molecule to harvest that energy, but it's not that simple, right? To break it apart, we have to add some energy. So even though that molecule's got energy stored into it, we have to sort of crack it open to get the energy. It's just not going to come out on its own. We need to add energy to break those bonds. We need to stress those bonds to start pulling those atoms apart until that bond becomes so stressed it breaks. again, remember what this bond is. This is an electron that's shared by two atoms. All right, and so if we have, and it's going to stay that way, and then unless we start to move those nuclei apart and start to pull those atoms apart, now it's harder to share that electron because it's got farther to travel, or because the forces are messed up. This is the stuff that you would learn in chemistry, but we start to disrupt those forces that keep everything together, and so now it's harder to share that electron, and eventually we break that bond. Now, once we do stress that bond enough that it breaks, then all the energy that's stored in that bond is released, and we can capture that released energy and use it for something else. We can store it in a different molecule or something. We can convert it to kinetic energy. Okay, so again, we start with a certain amount of energy that's in the molecule, then we add some more energy. When you get to the peak, you've added so much energy, you start to break those bonds. But you had to put a certain amount of energy in order to break those bonds. Once you get to that point, you've added what's known as the activation energy. That's the energy that you have to go in until you break those bonds. When you break the bonds, it releases a bunch of energy. Now, when you get to that point, when you get where the point that I've got marked with the arrow here, at that point, you haven't gained anything. You've put in the activation energy. Now you're starting to get energy back out, but you've already invested some energy. And so at that point, you're only breaking even. There's no net gain in energy because you put in some energy and you got exactly that much energy back. However, there's more energy that was stored in the molecule. And so because that molecule broke apart, that molecule keeps releasing energy, and so that's the extra energy that you get from the molecule, and then you can use that extra energy to go do something or to store it in a different molecule or whatever. And so you first put in some energy, then it starts to release a tremendous amount of energy, but at some point you only get back what you put in, but then you get back a little bit more. And that's why it's worth it. That's why it's worth to put that energy in at the first place, because you're going to end up getting more back because there's energy stored in that molecule. And that's what this whole lecture is about. This whole lecture is about cellular respiration, getting at that energy that's stored in the molecule. So another way to talk about this is we say you're priming the pump. So if you've ever used a pump, sometimes you have to prime it. You have to put some water into it to get the pump to work. That's what priming the pump is. This is what we're talking about here. You've got to put a little energy in to get a lot back out. Why do you need to put that energy in? You need to stress those bonds to the point where they break. And then when they break, they release a tremendous amount of energy. And so again, that's called the activation energy. The energy you've got to put in to break those bonds. Okay, now we're going to talk about enzymes a little bit. So this molecule that we're working on is pretty stable. Most molecules are at a fairly low energy state, so they're not going to break apart on their own. Eventually, this molecule would break apart on its own if we waited long enough. That's what the second law of thermodynamics. The second law of thermodynamics says everything tends to disorder. And so this molecule is kind of an ordered molecule. Eventually, it's going to decay and break into simpler components if we wait long enough. But that's going to take a long, long time. And living things can't wait that long. We just ate this molecule. We need its energy now. If we don't get that energy, we're not going to be able to keep up our life processes. And so what we need to do is we need to speed up that reaction. We can't wait for that molecule to break apart on its own. We need to make that reaction go faster. And so how can we speed up a chemical reaction? So if you've got any kind of general chemical reaction, what's an easy way to speed it up? An easy way to speed it up is to just heat it, right? And so anytime you heat a chemical reaction, the molecules are going to be flying around faster, and that reaction is going to speed up. But this doesn't really work for living things, right? That's a bad idea. So if you want reactions to go faster in your cells, you can't heat your cells up. You know why, right? You know that your proteins will denature, and that's going to be a problem, and things like that. So that's not going to be a good strategy for speeding up this reaction. A better strategy is to use a catalyst. And so a catalyst is any type of molecule or anything that speeds up a reaction but itself is not changed. And so, you know, it doesn't have to be a chemical. There's lots of things that can act as a catalyst. But in biology, a biological catalyst is called an enzyme. And so you've probably heard of enzymes, you're probably familiar with enzymes, but this is specifically what an enzyme does. An enzyme speeds up a reaction, but the enzyme itself is not changed. And so now we're looking at our graph again, but this time we've got a new line. And this new line is the energy that it takes to break this molecule apart using an enzyme. And what the enzyme does is it lowers that energy of activation. You still need to put some energy into the bond, into the molecule to break it apart. You still need to add the energy of activation. But with the enzyme, you don't have to add as much energy. And so if you look at this, the red line here, you see that it does go up. You do add some energy, but then very quickly those bonds break and then it releases all the energy that was stored in the molecule. And so that's what an enzyme does. And in this way, it speeds up these reactions so that they happen faster, so that you can use that energy. And that's what you need. You know, these reactions need to happen faster so that you can stay alive. And so again, both lines represent the same reaction. It's taking this molecule AB and breaking it into A and B and releasing the energy. But the enzyme means you have to put way less energy in to break those bonds. And so the reaction is faster, it's much more likely to happen. So here's a figure from your book, just showing you kind of a general diagram of an enzyme. And so you've got this big molecule, which is going to be a protein. You remember proteins are big and they've got funky shape, and it's got an active site. And then the molecule it works on is going to fit into that active site, and the enzyme is going to catalyze a reaction on that substrate. And the enzyme itself is not going to be changed. So let's illustrate this with an example. So here's a sucrose molecule, very simple carbohydrate, and this is something that you could ingest, and you want to break it apart to harvest the energy that's in this sucrose molecule. We need this molecule to break up. We need this reaction to speed up so we can use the sucrose is just sitting there. We need it to break apart. Well, you could heat it up. You could add heat, and then that sucrose would break apart. But of course, that's not going to work. You can't heat up your cell this way to make this thing break apart. We could just wait. You could wait 100 years, 1,000 years. The second law of thermodynamics says eventually this sucrose molecule is just going to break apart on its own. But that's going to take forever. That's not going to work either. So what we'll do is we'll add an enzyme. And the enzyme that we're going to use is called sucrase. And so this is something you're going to notice, that anything that ends in ASE, anything that's called something ACE, that's probably an enzyme. So sucrase is an enzyme that works on sucrose. And so in this figure, what they're showing you is, again, you've got this big protein molecule with this very unique three-dimensional shape, but it's got an active site that perfectly fits sucrose. And so that sucrose can fit right into that active site. It's as if that enzyme was built for this purpose. And guess what? It was. This enzyme was built for this one reaction. No other molecules will fit into that active site, or few other molecules will. And so that's what we're saying here, that sucrase has an active site that perfectly fits sucrose. This is a protein Remember that proteins are all about shape. This protein's shape is designed for one specific purpose: to bind sucrose and break it apart. In proteins, shape is everything. Now, when the sucrose binds to this active site, the bonds in the sucrose get stressed. When the bonds in the sucrose get stressed, they break apart. When they break apart, that releases the energy. That was the goal all along. But instead of having to add heat to break those bonds apart, we just use this enzyme, and this enzyme stressed those bonds until they broke apart. And it took much less energy to break those bonds than if we'd have just heated it up. That's the advantage of using an enzyme. So we just split sucrose up and harvested its energy without heating things up. That's what we needed to do. Now the sucrase itself is not changed. So once it breaks apart one sucrose molecule, it can go find another, and then it can go find another, and it can just keep doing this. That's what a catalyst is. The catalyst itself does not get changed. Okay, now remember that DNA makes RNA, which makes protein. And this sucrase enzyme is a protein, which means that we can go and find the gene for this protein. And in fact, we know exactly where the gene for this protein is. It's found on chromosome number 3 in humans, and it's exactly between these base pairs. And so because we can sequence DNA now, we can find where these genes are, then we can study them in a lot more detail, and we're learning a lot more about biology because we can study things at the genetic level. Now you recall that a mutation is a change in the DNA. So we can go to any gene or any part of the DNA, and it can sometimes change. Now, if you change the DNA in a gene, you're definitely going to change the RNA. The DNA makes the RNA. You change the DNA, you change the RNA. Now, this might change the resulting protein, or it might not. And so, some mutations don't change the shape of the protein at all. And so the protein functions as it always did, even though there was a mutation. We call this a silent mutation. It's a change in the DNA that has no effect on the protein. However, the mutation might change the shape of the protein. But again, it's possible that the change in shape is not very important. And so you can change the shape of the protein and it still functions well. So that would also be a silent mutation. But it's possible that if you change the shape of the protein, now it doesn't work very well because, proteins, the shape of a protein is tightly tied to its function. And so if you have a mutation that changes the shape of the protein, that's very likely to cause that protein to not work. But it's also possible that that protein can work a little bit better. And it doesn't have to be just enzymes, it could be any kind of protein. And so the take-home message here is that you can have mutations that might not do anything, or they might do something very harmful, or they might do something that's very good. It all depends. And so we've studied mutations in this sucrase gene, and it's also called the sucrase isomaltase gene. So, it can work on a couple of very similar molecules. But researchers have found that a mutation in this particular gene makes people unable to digest sucrose. And so you've got a change in the DNA, it changes the shape of that enzyme, and so the enzyme does not bind to sucrose very well. And so if you don't bind to sucrose, you can't break it apart. And if you can't break it apart, you can't digest it. And some of the side effects of this are stomach cramps and bloating, gas, diarrhea. And if you study this and you look at people's ancestry, you can see that about 1 in 5,000 people from European descent have this mutation, but it's much higher if you're from Greenland, Alaska, or Canada. You've got a much higher rate of this mutation. Well, being unable to digest sucrose and if you eat sucrose, it gives you gas and it gives you diarrhea. Does that sound like anything else you might be more familiar with? Has anyone ever heard of lactose intolerance? Well, sucrose intolerance is very similar to lactose intolerance, and for a very similar reason. And so lactose is a different kind of carbohydrate, and so we have a different enzyme that breaks down lactose, and it's called lactase. But in the same way, it binds to lactose and breaks it apart. Now, there's been mutations identified in this gene, and some mutations cause an amino acid chain in the lactase protein. And so again, you've got to change the DNA, it changes the RNA, and it changes the RNA in such a way that changes an amino acid. It changes the primary structure of that lactase protein. That changes the shape enough so that the lactase does not work. And so there's a couple of different mutations that cause lactose intolerance. If you have lactose intolerance in babies, it's often caused by a mutation to where the shape of the protein is changed and it just can't break down the lactose. If you have adults that are lactose intolerant, it's sometimes a different mutation, and it's a mutation in which the DNA is not transcribed as often as it should. So when the DNA makes the RNA, that's called transcription, and there are genes that control how fast other genes are being transcribed. And adults with lactose intolerant often don't make enough of this RNA, so they don't make enough of the lactase. But again, by understanding the genetics and understanding the process, you can understand some of these disorders. Okay, last thing we need to talk about in this section is making ATP. So we've been talking about breaking apart molecules to get their energy. Now we need to stick that energy somewhere so that we can use it, and that somewhere is usually ATP. And so sometimes you say ATP is sort of like a battery in the cell. You charge it up, it carries energy to somewhere else, and then you use that energy. You need to move energy from place to place, and ATP is your number one way of doing this. So ATP stands for adenosine triphosphate, and it's a nucleotide. And so, adenine is one of the nucleotides you learned about that's used to build DNA. This is actually a ribonucleic nucleotide, so this uses ribose. And so here's a figure from your book showing the structure of ATP. And you'll notice, you recall what a nucleotide is. It's a sugar with a nitrogenous base and a phosphate. And so if you look at this figure, you see you've got a pentose sugar, which in this case is ribose. You've got a nitrogenous base. You've got the adenine nitrogenous base, just like you used, in RNA before. But instead of having one phosphate, now you have three phosphates. And so that's what makes this different. Those three phosphates have lots of energy. And so what they're showing you here is those two extra phosphates are held on by these very high energy bonds. And so where does that energy come from? It comes from your food. What we just talked about, when you break apart a molecule and release that energy, you can use that energy to stick on these phosphates. Now those phosphates hold that extra energy. Then if you break off those phosphates, you release all that energy. So you can take and use ATP to carry energy from one place to another. Here's another diagram, again showing you the adenine nitrogenous base attached to a ribose, which is a pentose sugar, and then it has three phosphates on it. You remember we talked about phosphate before. A phosphate is a phosphorus with 4 oxygens attached to it, and it's got a negative charge. So now you're jamming together 3 of these big molecules with that negative charge. Negative charges repel. They don't want to come together. To get them to come together, you've got to really put a lot of energy into it until they get so close that they start sharing electrons and they're held together. And so That's why these are high energy bonds, because those phosphates don't want to come together, but we use energy to force them together until they bond together. And so all that energy we put in to bring them together is now stored in that bond. Consequently, if you break that bond, you release a tremendous amount of energy, which is what you want ATP to do. And so what are some examples of how we use this? Well, let's go back to our earlier example of sucrase. You remember we said that sucrose binds to sucrase, and then sucrase stresses those bonds to break the sucrose in half, right? Well, how does it stress those bonds? How is it able to do that? Well, it uses an ATP. So once the sucrose binds to the sucrase, then an ATP comes along and transfers energy to the enzyme. This energy causes the enzyme to change shape. When it changes shape, that's what stresses the bond. And so the enzyme takes energy from ATP, changes shape, stresses that sucrose bond, and that breaks the bond and releases the energy in sucrose. So again, you can see why, the analogy is ATP is like a battery that carries energy from one place to another. So this ATP went and picked up energy from somewhere else in the cell, carried it to the sucrase enzyme, transferred energy to the sucrase enzyme so that the sucrase enzyme could break apart the sucrose. Now, just a little digression here. It's something you're going to see on a lot of these slides. You show in the diagram here, it shows the ATP is converted into ADP plus P. And so that's a standard way of writing this. And so the ADP is adenosine diphosphate. And so it's only got 2 phosphates. And then you can see that it's ADP plus P. So there's that 3rd phosphate. What they're trying to show you here is, you've got ATP that's got 3 phosphates. One gets broken off. The energy that held that one phosphate is released and transferred to something else. And so you're going to be seeing this in a lot of our diagrams. And this is how ATP transfers energy from one molecule to another, by releasing that high energy phosphate. And so then the energy that's released can be used. And then later, we'll see as we talk about cellular respiration, we'll reverse this process. We'll use energy from your food to stick a phosphate onto an ADP, and that's how we'll make ATP. Okay, so remember when we talked about, enzyme kinematics and that, we said we need to prime the pump. We need to add a little energy in order to release a lot of energy. And so this is an example of how we do that. We add a little bit of energy from that ATP, but that energy is very, it's used in a very efficient manner. It's used to change the shape of this enzyme, which breaks that bond. Again, we could add a lot of energy and heat it up, and that would break the sucrose bond. That's not going to work in living things, right? That would kill the organism. By using ATP in an enzyme, it's sort of like, using a lever, right? You're using the energy in a more efficient manner. And so we're able to use just a little bit of energy from ATP to change the shape of the enzyme, to stress those bonds, to break them, to release the energy. And then we can use that energy for something else. Okay, so that's part 1. That's just an introduction into a lot of the concepts that we talk about when we talk about cellular respiration. In part 2, we're going to get into some more of the details about how we're going to actually build, charge up these ATPs. So let me know if you got any questions, and I'll see you in part 2. Later.